I'm happy to introduce Thorsten Wagner. So he's here to talk, joining us today from somewhere over the internet that I believe is nearby Max-Planck-Institute in Dortmund, and he's here to talk about crYOLO. So with that, I will turn it over to him. And Thorsten, thanks for joining us.
Thanks for the invitation. So I'm going to talk about, yes, crYOLO, but also its utilization and feedback-guided real-time processing. And yeah, so the presentation is basically split in two parts. First, I introduce crYOLO again. And the second part will be about the integration of crYOLO into Transfire, which is a software for automatic processing. And yeah, I will highlight how crYOLO is used in Transfire. All right, then let's start with crYOLO. So particle picking is in principle nothing more than common object detection. And one of the very common approaches is to train a classifier on positive, like the particle, and negative, like contamination and background examples. And then you use this classifier and it is convolved over the image. And each of these small blue windows is cropped out of the image, is presented to the classifier, and the classifier will give you some probability that this image contains the object that you want to detect. And in the end, you get a kind of probability map, and where you detect the peaks in such a map, and these peaks are the position of the particles. This procedure is computationally expensive because you always have to crop out the image and pass it through, let's say, a convolutional neural network. This takes time, and these blue boxes are overlapping highly. And moreover, if you use such a small window, such a small cropped out region, you have very little context. So in crYOLO, we are using an alternative approach called you only look once, or short YOLO. And YOLO does the detection in a single run through the network by passing the complete image into the network. And then it uses a grid to detect the objects, in our case, the particles. And if we compare it, the two approaches, a common, let's say, common sliding window approach and the YOLO approach, then for the training data, the good point is you don't have to select background and contamination. It's enough to select the particles. Because the network always sees a complete micrograph, it has a much bigger context. And in terms of computational efficiency, this is also very good because, yeah, instead of putting all of these small cropped out regions through the network, you can just put once a complete micrograph through the network and you get your detection immediately. So a little bit of technical background. So to use crYOLO, you typically label a small set of micrographs, let's say 5 to 15. And then they are passed multiple times through a convolutional neural network. And the result of the last layer is a stack of grids. And each grid cell itself represents a small region in the input image. So the grid cells of the first two layers basically answer the questions, is there a particle? And if yes, what is the confidence of seeing a particle? The third and the fourth layer are estimating the relative X, Y positions inside the grid cell. And the last two layers estimating the width and the height of the box. And this is the function that is optimized during training. It's a so-called loss function, and it has 4 parts. the first part, by the way, can you see my mouse cursor? I hope so.
I can see your mouse fine.
Okay, good. So the first part is penalizing the estimated errors in the estimated position. The second part is penalizing errors with the estimation of the width and the height of the boxes. And the third and the fourth part is penalizing errors in the confidence. So boxes that contain the center of a particle should show a confidence of 1, and grid cells that don't contain the center of a particle should have a confidence of 0. And then you have these weights in front of each term. And in crYOLO, we put a much higher weight on errors that if crYOLO does not pick a particle that you have selected. Okay, that's enough technical details for now. Let's see how you would use crYOLO in a practical way. So this is the GUI of crYOLO, and you have different actions here, and you have tabs with options. And I would say in 99% of the cases, just the first tab is relevant for you. So here we have selected the action config, which creates a configuration file. You specify the name of your configuration file, the box size you use, the folder where your training images are, and a folder where you have your annotation files. So, and these are created by a separate tool of crYOLO. It's called Box Manager, and I will introduce it later. And you specify where do you want to save your trained model. And if you press start, you get this a JSON file with this configuration here. You see there are more entries that are shown in the GUI here, and these entries are basically hidden behind the other option tabs here. For example, the validation image folder and validation annotation folder, it's empty here because crYOLO typically selects the validation data randomly from your training data. So it takes 20% of your training images as validation images. That means these training images are not shown during training. They're just used to measure how good is crYOLO right now for this data set. And if the performance measures do not increase anymore on the validation data, then crYOLO stops the training. But in principle, it can be specified in the validation configuration tab, and then these two lines here would be filled. Okay. Next action is training. You specify your configuration file and the number of the so-called warm-up epochs. The default value is fine. You can use 5 in basically all projects. What does this warm-up epochs mean? It means that in the first 5 epochs of training, it will ignore more or less the estimation of the width and the height. Why do we do that? Because in the paper of YOLO, there's written that this helps the network to converge, and that's the complete reason. So, and if you press start, after a few minutes, in this case 7.5 something minutes, you get a model for picking. The time it needs for training is dependent on the number of training images you provide. In this case, it should be 14 images, I guess. Okay, and with your model, you can then start the actual picking with the predict action. Therefore, you specify again your configuration file. You select the model that you want to use. You specify where your micrographs are and where do you want to write the new box files? Then you press start, and with roughly 6 micrographs per second, it will produce box files for you. It will produce star files, emen2 box files, and the so-called cbox files. And one folder called distribution. So this folder here, it contains information about the confidence distribution. So each particle that is picked has a confidence ranging from, I don't know, 0 to 1. And the distribution folder contains the distribution of this confidence value. And the cbox files, they contain for each particle the confidence itself. And this can be used inside the box manager. So if you start the box manager, you can also start it from the GUI. Then you load your micrographs and the corresponding cbox files. And it might be that you are not happy with the default confidence threshold, which is 0.3, and you want to adjust this threshold. So you load your cbox files, and then you can increase or decrease the threshold depending what you want to achieve. Maybe Criollo picked some contaminations, and it might make sense to increase the confidence threshold, or it misses some particles, then you might want to decrease the confidence threshold. If you are done, you simply save your new selected particles into a new folder, and it will write emen box files and star files. Besides the visual inspection of your picking result, you can also calculate numbers that give you an idea how good the picking worked. And for this, we have the action evaluation. Again, you need to specify your configuration file and your model, and then a so-called run file. Every time you run crYOLO, it creates a run file in the logs folder. And this run file, it's a JSON file, contains the information which images were used for training and which one were used for validation. And then you can calculate based on the validation images a set of numbers like precision and recall, and the summary statistic here, the area under the curves, it gives you an idea how good your picking was. So as high as this number, it goes from 0 to 1, and 1 is the best and 0 is the worst. Yeah, you have an idea how good your picking was in terms of your validation data. And it will also tell you what might be an ideal picking threshold. And this is basically evaluated by the F1 or F2 score, which is nothing else than a weighted mean of precision and recall. But the F2 score gives a little bit higher weights on the recall, which might be the more usual case or more important metric for cryo-EM. Okay, now let's look at a couple of examples. What I really like about Criollo is that with the configuration that I just explained to you, you could have trained all sorts of projects. You could have trained a model for picking very big particles like these viruses here. Oh, wait, these viruses. Or a very crowded example like here. Or you might want to train a model that picks particles very specifically, like here the model picked HIV spikes that were attached to liposomes, or a similar example where it picked MPGs to liposomes, attached to liposomes. If your training data also shows contamination, Criollo will learn not to pick this contamination. So it will ignore it. And for some data sets, it might be hard to create the training data. So you might miss particles during creating the training data. But Criollo is pretty robust against sparse picking. So, and to demonstrate that, what we did is, this is our TC toxin data set, and the training set has 10 micrographs with 1,100 particles. And what we did was we removed randomly 80% of the boxes. And what's left are 205 particles in 10 micrographs. And then we used this sparsely picked training set and trained a model, a CRYOLO model on it. And if you then apply CRYOLO, you see that it misses quite a lot particles. But if you then use the C-box files and reduce the threshold a bit to 0.14, you get basically all your particles back. And we repeated this experiment on our TRPC4 data set. So basically, we did exactly the same. We removed 80% of the training data and trained a model on it. And if you run then the picking with the default threshold, it misses some particles. And if you reduce it to the lower threshold, you get your particles back. And the next thing I would like to show you is the possibility to use general models. So you might have multiple data collections of the same particle with different settings, different grids, a different camera or another microscope. And a model trained on the data of one data collection might not perform optimally on a data set from another data collection. And to get better generalization, what you can do is you can just merge training data of multiple data sets by putting them in separate folders. And what you get is a model that generalizes better and also works with data of data acquisitions from totally unseen, on unseen grids or something else. And if you have enough particles, types of particles in your training set, you get a real general model. So, and we did that for more than 60 data sets, and we trained CRYOLO on it. And what we get is a model that you can apply to new data sets that are not contained in the training data. And on the right, I put a couple of examples. And the good thing is, if you train CRYOLO on many different particles, you get also an estimation of the size if you apply CRYOLO on new particles. So this is shown here. I load again the C-box files. And now I show you the confidence distribution and the size distribution. And now it might be, okay, let's first adjust the confidence distribution a bit. And now you can use the estimated size. You see the boxes are now bigger and smaller. And you see two peaks in the particle diameter distribution, and we want to remove the bigger ones. Those particles that obviously are in this bigger region of the ice. And we can just change it by setting the minimum and maximum size. And this allows you to select a subpopulation in your data set. The last feature I would like to show you is the filament mode of CRYOLO. So training a model for filaments is in principle the same as training it for single particles. The only thing that is different is that you create your training data with, for example, the E2 helix boxer that is contained in the EMN2 package, and not with the box manager. Then you train your model as shown before. And if you do the picking, then this works a little bit different. So first, CRYOLO will pick the individual boxes, but at the same time, it will convolve the input image with this kind of mask with this kind of kernel. It will rotate the kernel, and using this approach, we can estimate the direction of the filament at every position. And then what it does, it chooses a random box and it searches for the next box in the direction it gets from this directional image. And it limits the search range by this angle alpha, and also by some distance, which is proportional to the box size. And in the end, you have traced filaments. And if we look at a couple of examples, here, for example, team B, this is the manual selection. And this is the result of crYOLO. Then you see that crYOLO is able to reproduce pretty much what the user had selected before. So you see in the manual selection by the user that he basically skipped those filaments that are too close. And crYOLO was able to learn that and to replicate it in principle. And the same for the F-actin. You see that no crossings or almost no crossings are picked. And also the filaments on the carbon edge are not picked. To summarize the crYOLO part, the crYOLO is fast. You can run it with up to 6 micrographs per second. With the context, it's kind of smart. The training is easy because you only have to select a couple of micrographs, and often it's not needed because the general model is working already for you. And even if you are not able to select all of the particles during training, it's quite robust against sparse picking and will still work. In case you are interested in crYOLO, go to our documentation website. It contains an extensive user guide with several tutorials, how to use a general model, how to train a model for your own data, how to fine-tune a model that was not shown in this presentation today, and how to use the filament mode and so on. It has background information about the networks that, or the networks that we are using. And take a look if you're interested. So that brings me to the next part of this presentation, the integration of crYOLO into Transfire. So Transfire is a tool for automated processing, and it's developed in our group by Marco Staprin. It not only takes care about the common pre-processing steps, it also provides you all sort of metrics during data acquisition. But Transfire goes a little bit further. So it's also calculating 2D classes and 3D reconstructions while you are doing the data acquisition. And this everything happens in parallel. So after roughly, for this case, it was TRPC4. After 1.4 hours, you already get results from the 2D classification. The feedback loop, and this is what I'm going to talk about next, is finished after 7 hours. And then you get your first reconstruction, the first reconstruction after less than 24 hours. But the calculation of the 2D classes and the 3D reconstruction requires a good picking. And this is automatically generated using this feedback loop. And this is what I want to talk about now. So the feedback loop is pretty simple. So we start our particle picking, we do 2D clustering, and then we select the good classes and provide the new, or the positions of the particles in those good classes. We provide it to crYOLO. It will retrain its model, and we do that again. We do that a couple of rounds until a user set number of rounds are done. And this is only possible because we developed two tools in our group. One is a GPU accelerated version of ISAC, and you can find, this is a 2D classification tool, and you can find it on our website. And the second tool is Cinderella, which is a tool to automatically select good or bad classes. Cinderella is pre-trained on ISAC classes, and we use that in our feedback loop. So let's look at a couple of examples. So to demonstrate the feedback loop, that the feedback loop is able to recover from really bad initial picking, what we did was we picked with a general model of crYOLO, and then we replaced 90% of the boxes with just random picks. We just put them somewhere on the image. And then the feedback starts. So the particle extraction, and then we do the 2D clustering. We select the good classes, generate box files out of them, and retrain crYOLO, which gives a new model, and we do the particle picking again, and we do that for 5 to 6 feedback rounds. And what you see here is that after 5 rounds of feedback, you get a pretty decent model that you can use for further processing. And also the statistics are quite impressive. So initially we start with less than 10 particles, good particles, per micrograph. And after a couple of rounds of feedbacks, we are getting more than 60 good particles per micrograph. And when I say good, I mean good in the sense of Cinderella classified it as good. And if we then use those good particles to calculate a reconstruction, we also get a pretty good resolution on 500 micrographs. So with 30,000 particles, we get a 3.6 angstrom structure. This also works for filaments. The difference here is that for filaments, we don't have a general model available. So what we did was we used a model that was trained to pick F-actin data, and we used that as initial starting point for this actin-myosin complex. And you already seen the images that the actin-myosin complex looks quite different from the F-actin data, and also the result of the picking is poor. There are some filaments are picked, but most of them are skipped. And after picking after six feedback rounds, you can see that we have a good tracing. It's not perfect yet, but it's enough to get a good reconstruction. And we see the same pattern again. We start by with very low number of good particles per micrograph, and this number is increasing. And in the end, we get a reconstruction with 4.4 or 4.4 angstrom with 100 micrographs. To summarize that, Transfire can be used for fully automated processing. It's complete. It's not only containing all of the pre-processing steps, it also contains 2D clustering and 3D refinement. And all of this is done in parallel. Additionally, it shows you various metrics. You have basically a live monitor of your data acquisition, and it's able to use feedback provided by our feedback loop to get 3D reconstructions and 2D classes for data sets that are maybe not picked very well by the general model by using our feedback loop. And with that, I would like to thank you for your attention. And of course, I would like to thank our head of the group, Stefan Raunser, and the complete Swire team, and of course, the complete Raunser group. Thank you.
Thanks, Thorsten. That was a very good talk. So again, for folks who have questions in the audience, feel free to send them to me by chat and we'll unmute you and you can ask. One question that I have, and this is possibly more machine learning related than cryo-EM related, but you talked about some of the different training approaches that you used for crYOLO.
Mm-hmm.
Have your talk about that and the confidence intervals reminded me of active learning models as kind of a halfway between unsupervised learning and supervised learning training. I'm wondering, is that something that has been looked at and is just the transfer learning with the generalized models is a better approach, or is that something that wouldn't make sense to try to investigate there?
I'm not totally sure if I got your idea. So what is the idea you want to do?
This is an approach where during training, a classifier will use the confidence to pick examples that it is most uncertain about, and then ask an experimenter to validate, is this a positive example or a negative example?
I see. So constantly feedback by the user.
Yeah, basically. More in a batch mode sense. It does sound very similar to your other discussions with the feedback loop during Transfire.
Yeah. So in principle, you want to replace Cinderella by a user, where the user decides, this is a good one and this is a bad one, a bad class, or directly in cryo and not in the feedback loop.
I've been thinking more about the Criollo training rather than the Transfire training.
I didn't thought about such a approach yet, because I would imagine that this is quite intensive for the user. But in principle, if that I could imagine that this will gonna work, but yeah, I have to see how intensive this would be for the user and if it's doable. But sure, I don't see any reason why it should not work.
It's more of a general question than a feature request. So
yeah, I see.
So with that, we have a question from Bharat Reddy, and so I will unmute you as soon as I manage the mechanics there.
Hi, I find your pipeline very interesting. How does it deal with heterogeneous particles, and can it separate different conformations while running this automated pipeline?
So right now in Transfire, everything is integrated until 3D refinement, including 3D refinement. In principle, it's also possible to use our tool for 3D clustering to separate specific conformations. And we have one example that I did not show here in the presentation, where we used our feedback loop for a data set of the holotoxin. And sometimes you have the toxin in the pore state, and sometimes you have it in the pre-pore state. And we prepared a model that basically just selects the pre-pore state. And in the end, we get a model that is pretty good on picking just those particles and not the other conformation. But of course, sometimes it's very hard, because the top views, for example, look very, very similar for this protein. But so yes, you can approach these heterogeneous data sets, but you probably would have to provide a Cinderella model that knows what is the conformation you are looking for.
So our next question from Michael Fenwick. So Michael, just a moment while I track you down in our list. And you should be good, Michael.
Thank you. I just would like to know the effect of the auto-picking program picking partial particles.
What do you mean by partial particles?
So it's not included in what's picked is actually not a full particle. based on the.
I'm not sure if I got the question. So,
just to try to clarify, are you talking, Michael, about a case where a particle is spanning the box boundaries, or a case where the particle is, okay, not the case where the particle is outside the image itself?
Right.
I see. So, of course, so one option is, as the general model of CRYOLO already estimates the size of the particle, you could use this information to choose the correct box size. This would be one option, or it has to be user provided. So either the estimation of the box size is correct, and I mean you can multiply the estimated box size with some safety value of 1 point something, or the box size has to be provided by the user.
Yeah, great. Next week we have a question from Mike Eck.
All better. So yeah, the iterative particle picking is just brilliant, but I wonder if there's a danger of losing rare views, and how do you avoid that? Or is it not an issue at all?
So yes, we also had this in mind, and therefore, if we start with our initial picking, we choose a very, very low confidence threshold that we basically pick everything what is there. And then we just rely on ISAC that it puts the bad particles into a contamination class or something else. And in the end, after we did a couple of feedback rounds, then we use our evaluation script to get a better threshold, and not to use this very low threshold that might also pick contamination. So yes, this is our strategy to avoid this issue.
Yeah, perfect, thanks.
Good, all right.
All right. Those are all the questions that I've seen so far. So I guess, unless anybody else has any last minute ones.
Maybe one last comment that I forgot to mention. Transfire in this, if you look up Transfire right now, the features that I described are not available in the most recent version. If you're interested in giving it a try, just contact us and we can provide you our current beta version. Yeah, this is just one important thing I would like to note here.
Great. Well, thank you again for presenting today, and thanks everybody in the audience for showing up. And have a good afternoon and rest of your other day.
Thank you.
Thanks for listening.